Okay, the next panel is on autism, molecules to minds. Our moderator is Jaideep Kapur. Kevin Pelphrey is on the panel, Micah Mazurek, Adema, Ribic, and Jack Van Horn. Jaideep, take it away.
Welcome. My name is Jaideep Kapur. I'm a neuroscientist and a neurologist. I do epilepsy research on mice and men, both, yes. But biggest thing is that I'm a graduate of UVA Neuroscience program. Let's do introductions first. Start with Adema.
Hi everyone. Can everyone hear me? Yes, wonderful. Thank you. It's so great to see everyone here. My name is Adema, Ribic, and by training, I'm a molecular biologist. I was also born in Bosnia, so I firsthand saw how traumatic experiences during childhood can actually change brain development trajectories. And I enrolled in neuroscience for my PhD in Germany, and then I arrived here to do my postdoc and kind of threw myself into developmental neuroscience. Kevin?
I'm Kevin Pelphrey. I'm a developmental cognitive neuroscientist. I study developing brain from fetal development into early aging. And let's see, I started my educational journey at North Carolina State, then to UNC, and then to Duke. So I am thoroughly confused about who to root for in basketball games.
Good day, everybody. I'm Jack Van Horn. I'm a professor in the Department of Psychology as well as in the School of Data Science. I did my undergraduate work at Eastern Washington University in Washington State on the other side of the country. I went to do my PhD work at the University of London in England, where I was homeless and lived in my office for several months because I had nowhere else to go. And then I did a postdoctoral fellowship at the National Institutes of Health, where I simultaneously undertook a master's degree in engineering. And I've been around at various parts of the country at various institutions and very happy to be here at UVA since 2019.
Hi everyone, I'm Micah Mazurek. I'm a clinical psychologist by training and I'm a professor in the School of Education and Human Development. I've been at UVA since 2017 and have been working on autism since that time. My work focuses on assessment, treatment, intervention across the lifespan, and dissemination into communities.
All right, so we'll get started. I think our panel was set up by a question in the by Aminata, who said, what about atypical development? You've been talking about disability versus normal development as though there is no atypical development. Actually, autism is not a disorder. It's just atypical disorder, atypical development of the brain where people have differing abilities, and the challenge of autism is to recognize the differing abilities of a child and to use them so that they can function effectively and contribute significantly to the society. With that, I'll start with Adema, who will talk about circuits again, but circuits in context of development of autism and atypical development.
This is with the risk of sounding very repetitive because a lot of what I will talk about is what the panel already introduced before us. And I will also mention Howard and say that I'm not a clinician, but I'm a friend, I'm an aunt, I'm a cousin with young parents who have children, and I have never seen anything more terrifying to parents of young children than someone telling them, hey, your child is diagnosed with autism. It's something that you can't detect early in blood work. You cannot image it with a PET scan. You simply cannot predict when it's gonna occur, how is it gonna occur. And autism is a very complex neurodevelopmental condition that manifests in very atypical social behavior, very typical communication, very typical sensory processing, and we still don't know the root causes of it. So one of the challenges of our field has been, for the past few decades, is to understand how autism is caused, how it arises. And it has been a very difficult challenge to address. I'm gonna say that from our end, we have identified around 400 genes that predispose a person for a high risk to develop autism. We have also identified a dozen neurodevelopmental factors, external factors such as maternal infections, for example, preterm birth, that again predispose a person to develop autism. We still don't know how a single one of these 400 genes results in a behavior that is very typical of autism. We don't know how a single gene leads to development of connections between neurons that will determine how those repetitive behaviors arise. Why are some children displaying them and why some not? How can all those 400 genes lead to the exact same complex disorder? And this gets to the gist of the problem. We've been trying to do this in a very simple reductionist way. We have been knocking out a gene, studying its function, studying its effects, and now we have 400 mouse mutants and we still don't know what causes autism. We have tried to change different environments, and we have infected pregnant mouse moms to study their children. We have studied many different things, and we still don't know if that's the root cause of autism. And for us, I mean, my lab works on that. We study how these different connections between neurons in the brain form, how they develop, how they change with experience. We study genes that build these synapses. We study how they actually structurally organize them. And one of the mice that we have in the lab is also linked, the mutations in this gene are linked to autism, but these mice do show atypical social behaviors, but they're also terribly smart. They learn new tasks quicker than any mice that we have ever seen. On the other hand, we have mice that show the exact same social deficits, but they also show learning impediments and repetitive behaviors. And this really gets to the gist of the problem of autism. It's a very heterogeneous disorder, not just in terms of its manifestations, but also in terms of its causality. So all of these genes and all of these factors are involved in its development. And for us neuroscientists, I think it's really key to understand how these synapses give rise to circuits and these different behaviors. How can a single gene build a synapse in such a way to give rise to normal speech and to give rise to atypical speech? How can these genes build circuits that give rise to neurotypical behaviors and neurodivergent behaviors? We still don't know that. And in my field, this is the biggest challenge. Once we understand how those things happen, we will be able to fine-tune the circuits, fine-tune the behaviors. One thing that we would be able to achieve by knowing that is to simply identify these really key time points during development where we can intervene. And JC talked about it, Sarah talked about it, Mark talked about it. So there are these very, very distinct windows of development during our childhood where we simply both develop and exhibit certain functions. Children start to speak around a year or two old. We still don't know how. Why? What happens in their brain that turns on and that they simply start vocalizing? What happens in the brain of an autistic child that doesn't turn on or turns on twice that they vocalize differently? We simply still don't know that. And the big thing, for example, another big problem is translation. We have been working in silos for a really long time, separately from clinicians, separately from developmental psychologists, and we need to breach that barrier to really translate findings like ours to clinic. My lab, like I mentioned, we study synapse development. We study how perinatal trauma, such as preterm birth, affects development of visual function and development of cognitive abilities. And we recently found and we're piloting a new study to pilot an intervention in the NICU to improve outcomes of preterm children. But we need more people like that, and we need better connections between people like me and people like Dr. Pelphrey and Dr. Micah Mazurek. So how do we build that connection between fundamental neuroscientists like me and like them? And I think UVA is at a great spot for that. I joined UVA because of such great community. We have people like me, fundamental neuroscientists like myself, like JC, like Sarah, like Mark, like Ao, like John Lukins, like John Campbell, like Barry Condra, like Igor Promazzi. We have all of us here who can really inform the clinicians and the developmental psychologists where to take, how to treat, and how to help these children. Because in the end, we really can't do that. We can't tell them, oh, hey, don't do this treatment because it doesn't really fit genetically with this profile, or this genetic profile may be better fit for this type of intervention. And I think UVA is really, it's a big, good place for that. All of us are together on the grounds. We all know each other really well. We have departments at the med school like pediatrics and neurology that we closely collaborate with. We have, and on top of that all, to really top that off, we have School of Data Science, the only one in the country, that really permeates all of this and can connect all of these missing dots to one big whole picture. And I think this is really kind of our trip to the moon and back. And I don't think that there's a better time for that than now. Thank you.
Great. I guess 3 things I want to highlight. In thinking about this as a moonshot, you got a flavor of just how complicated autism is as a condition. What I think makes UVA particularly unique in being able to tackle that type of complexity. One is that we are actually already one of 9 autism centers of excellence in the world. And the other 8 are very, very well-known places that we are very proud to be considered among. And so we already have in place that sort of preeminence and that label for now close to a decade, hopefully 15 years. Grant renewal went out today. And the reason why I came to UVA, I had spent most of my career at the Yale Child Study Center, which is a pretty well-known place for the study of autism. And I ran one of the autism programs there, and that sentence should reveal one of the autism programs. There are multiple, and we're really competing against each other in order to go after this sort of great moonshot. And so for me, UVA is a terrific opportunity with the STAR program, which we'll hear more about, the Brain Institute, the School of Data Science, the Basic Scientists, which you've already heard from some today, as well as my colleague just now. That type of openness and collaboration and facilitation of team science and rewarding of team science, I've never encountered anything like it in any of the universities that I've trained in or been on the faculty of. And so I think that's something that we should be extremely, extremely proud of. And for me, I wear 2 hats. I'm also the parent of a daughter with autism. I think that's pretty well known. So, my moonshot, there's no saving anything for the trip back. The moonshot for a parent with autism is to get their child to adulthood and then figure out from there how to get them to Mars. And, how do we take this differently developing brain and optimize what it's good at, what it can do, and get it there. And I think that with the tools of modern cognitive neuroscience, combined with the types of things that Micah is studying for community implementation, Adema is studying in terms of circuit manipulation, and Jack will talk about in terms of data science, to quote the great astronaut Matt Damon, we can science the shit out of this. And really get to the point where today we given the resources to be able to work together, because none of this fits nicely in an NIH RFA, get together and go after this in a way that we're in the middle of it, trying out different interventions in human beings, first do no harm, but seeing how those interventions work and seeing what brain profiles combined with genetics, microbiome, immune function, actually predict for whom which intervention will work and when, and then from there develop new interventions. And I think that that's something that UVA can uniquely do because of all of the panels that you'll see today when you take them all together.
For me, the brain is a data science. You heard terms from Sarah and from Mark in the previous session, they're almost like Carl Sagan-esque, billions of neurons, trillions of connections. They weave this tapestry of connectivity that we look at across multiple spatial and temporal scales. The neurosciences do not collect less data. We only collect more data. You've heard about all that stuff. An effort such as the ones that you're going to hear described in this panel, as well as in the previous and future panels, these are huge. And in order to understand these at a very foundational level, we are going to collect probably more data collectively than an individual academic department probably collectively obtained in their scientific careers even a generation ago. It's that amount of data. There are a number of major efforts which go on now. We heard a little bit about some of the ones like the Allen Brain Institute, the folks at Genelia Farms, collecting these data just at like writ large. It's really quite amazing. There are several which have to do, that have been funded by the NIH, including the Adolescent Brain and Cognitive Development Study, so the so-called ABCD study, the Human Connectome Development Study, and importantly for our conversation here today is the one that Kevin mentioned about our Autism Centers of Excellence, which we are very fortunate to have here as one of several in the country. We're very fortunate about that. These are huge resources, and as Adema talked about, these are enormous resources for us to learn from, but also to help to contribute to. But where data sets range in size from terabytes to petabytes, to throw some other very large terms at you, things like moonshots almost seem cute by comparison, don't they? To put this into scale, the phone you hold in your hands is millions of times more powerful than the computers that NASA used to land men on the moon and bring them home to the Earth. When we have to have this kind of computational power at scale, we really need to amp up our game to be able to process the data that we're going to collect in the amount of time that we have before this decade is out, as it were. This is going to require contextualizing things in terms of new analytic workflows, new machine learning technologies, new algorithms. We're going to have to bring to bear all of these computing technologies to be able to use GPUs or massively paralleled CPUs to be able to process these data, bring them together and integrate them from genes to systems to phenomics, so that we can make connections from synapses to cells to systems to syndromes to societies. And with the kind of computational things we're talking about, we will be able to make models of autism spectrum disorder that can translate from the cell of the animal models to humans and involve new treatments and new ways of therapy. This is going to be hard. It's really going to be a difficult operation to bring that kind of computational power to bear on this. We heard about the different spatial and temporal scales that we would have to traverse. That is a difficult proposition. I think everybody in this room has probably chosen the brain because of that degree of scalarness across space and across time. For us to be able to take that computational challenge on, it's going to require some doing. But I think we've all would agree that we've chosen this particular challenge not because it's easy, but because it's hard.
Thanks, Jack. So I'm going to take us back to more of a high-level kind of practical approach. As a clinical psychologist, I'm thinking more about kids and families and individuals. And I just want to start with thinking about autism as a whole and following up on our opening comments about what autism is, for those of you who don't know much about it. It is a complex neurodevelopmental disorder that affects the way the brain develops from really early in life. We know that the prevalence of autism has increased rapidly over the past several decades, and it's increased by over 170% in the last 2 decades alone. So that means that 1 out of every 54 children in the US have autism. And what that means is that everybody sitting in this room has a connection to autism in some way, whether it's in your family or someone you know. The impact of autism is significant across almost every aspect of development. We're in thinking about social interactions, communication. It affects learning, behavior, and daily living skills. And so that means that individuals and families face really significant challenges across the lifespan from early childhood all the way into adulthood, as Kevin mentioned. So I think this is not only an issue of great societal importance for us to be thinking about, but it's also a true grand challenge if you think about things that are difficult. We don't yet know what causes autism. We don't know if a baby's going to go on to develop autism before their symptoms emerge and we can observe them. There's not a single treatment that works for everyone, and most communities have limited to no professionals or resources to help families. So we also know that people with autism bring really unique strengths and abilities. Their neurodivergent thinking could be capitalized on in a way that could help them achieve really positive outcomes. But most people on the spectrum are not achieving their true potential. So this is a grand challenge at the societal level and at the individual level when we think about individual lives. So as a clinical psychologist, my research focuses on real-world assessment tools, developing things that will allow clinicians to diagnose autism in their clinical practice, developing interventions to address both core features of autism, but also co-occurring conditions like sleep and anxiety problems. And I think also thinking about how to leverage technology to increase access to care for communities. So that's where my work kind of sits within this. But I've had the pleasure of collaborating with lots of you in the room across basic science to applied practice and projects focusing on these issues around the aspects of grand challenge for autism. And I think that as a field and as clinicians and educators, we're missing really critical developmental windows, and this came up in the earlier panel, for identifying the very earliest signs of autism before symptoms emerge, and we're not beginning interventions at a time when the brain is, poised to capitalize the most. So we're really missing the boat there. The age of diagnostic, of first diagnosis of autism is well beyond the time when we would want to start intervening for most kids. It's around, age 5, when we know that we want to start interventions in toddlers. So that's a big issue. The other big issue is that our currently available treatments were developed with a very minimal to incomplete understanding of the neurobiology. So they're not really optimally designed for benefit for most people. We're using a one-size-fits-all approach that doesn't work well for most people, and it doesn't work at all for some people with autism. So to truly move the needle forward in the field, we need, number one, faster, accurate, and objective ways to diagnose and identify the earliest signs of autism before the symptoms start to emerge. And number two, we need a better understanding of the underlying neurobiology and heterogeneity of autism in order to understand subtypes. We've all been using the term autism, but I think Adema's comments reflect the fact that it is not autism, it's autisms, and we as a field have no clue about how to define those subtypes, which has really held us back in a lot of really significant ways on the intervention side. So we need a better understanding of that biology and those subtypes in order to develop personalized, individualized treatments that are going to work for a specific person. So, the key barriers I think are common probably across all these panels thus far, and it's that we've been working in silos. Scientists and clinicians and educators are all doing their own thing, not talking to one another as much as we should be doing, and that's been a roadblock to discovery. So I think a truly interdisciplinary crosstalk from molecules to minds and back again would transform our understanding of autism and our ability to improve outcomes for people in the real world. I think our early investments at UVA in neuroscience and in autism have just been thrilling and amazing. We've made great strides in the field, and we're now poised to really capitalize on that momentum and address this bigger grand challenge that we're talking about here today. We need a seamless pathway for collaboration that allows this crosstalk that's bidirectional from science to practice and back again. Our science and laboratory science should be informed by real-world lived experience and insights from people in the community, educators and clinicians. And then those findings from the lab that are so exciting and groundbreaking should be more readily translated into interventions that can be implemented in the community. And then how they're working should come back to the lab to inform new insights from basic neuroscience. So tackling these challenges is going to require a lot of infrastructure and support for pulling in all this data that Jack talked about that's mind-boggling when you think about the scale of data that we need to really combine and integrate, but that's what is going to be necessary for us to move forward as a field.
So Micah, you began to talk about one grand challenge, which is specified treatment for subtypes of autisms. We know that there isn't one condition. So do you want to amplify a little bit on that, and then Kevin can pick up on that?
Sure. Yeah, so I think, one of the grand challenges the moonshot ideas, I guess, that we came up with was this idea around personalized treatment. To do that, we need a better understanding of heterogeneity and subtypes. and a better understanding of that neurobiology. And so that would then lead to more personalized interventions. And so that would allow us to decide very early what the best intervention is for a child at a particular point in time. So we're not wasting valuable time and developmental windows on things that are not going to work for that person. We want to create a personalized, individualized treatment plan and education plan for everyone. So this moonshot would require us to collect and integrate data from across disciplines, which hasn't really been done, I don't think, in our field. And this is what is really a moonshot, including genetics, biological information, things I don't even know about that could be on the biology side, brain imaging, all the way to things that I can do, which are more behavioral, phenotyping, cognitive functioning, social skills, and those kinds of things. Pulling it all together in a single, unified, centralized system that would allow data scientists and people who are great at computational modeling to help us understand those subtypes and follow trajectories, do deep learning, and other things that I won't pretend to really understand that well. But I think the ultimate goal for me is to have that information available to define targets for intervention, and then feeding the response to those interventions back into the system so that we can continue to learn and move forward. One thing that this would allow us to do is to use particular interventions at moments in time as experimental levers and probes to test our hypotheses around these subtypes and targets for intervention. So this would be a platform that could seed, hundreds and hundreds of clinical trials or other things that could spin out of this if we had this infrastructure in place to support that work.
Absolutely perfect. And I would add, let me try to flesh out a couple of examples. So, for instance, through our Autism Center of Excellence network, we've discovered that just when you look at biological sex, the brain systems involved on average in autism are different in boys versus girls. So just taking a snapshot and also looking at developmental trajectories, and who would have thought that? And these kids have received the gold standard diagnostic workup that has made them as homogeneous as possible. They're perfect research subjects, which is very different from real-world autisms. But nonetheless, when we look inside of their brains, they have different differences. And then when we look further, and then we use that information about brain development to inform where we should be looking for genetics, we discover that the genetics underlying autism in biological boys or biological girls appear to be different. In boys, it's what we always kind of thought it would be, something about synaptic development, circuit formation. In girls, the clusters of genes that come out have much more to do about early immune regulation. And so I get really excited thinking, boy, I could take this and talk to John Lukens, talk to EO, and really begin to understand developing therapies that could be broken into something as simple as biological sex. And that's just one example. Within that, we find that when we do find a very strong candidate gene in girls, it causes autism, epilepsy, and intellectual disability. And this is public knowledge, so I'm not sharing anything HIPAA-related. But there's a whole Simons Foundation-supported effort to study this gene because in male, it causes high levels of analytical thinking ability and extraordinary math ability. And so when passed from a father to a son, it tends to convey a beneficial effect. The son ends up going to elite private schools. When passed from a father to a daughter, it has a devastating effect. And so, precision medicine, we're at that threshold. And then to create animal models where we can really go in and understand why this is the case would be absolutely fascinating. And then the thing I think that is necessary in this, and it was brought up earlier, in a completely different context, I had the opportunity to visit the intensive care unit for neurology. Yeah, I'm a faculty member and neurologist, but I'm not a clinician, so I had never actually set foot in the place. And I was stunned, both in terms of being impressed, but also about what if we had a truly translational building where we're doing precision neuroscience, where we can bring in the data scientists, the individuals working on animal models, the people doing interventions, the brain imaging systems, and work hand in hand so that we're all kind of, and we're all glued together with data science, and how amazing that would be. And just to take that one step further, I think it's really important to point out that one of the reasons why Moonshot is such both a great idea and also controversial, so much of when we made the Moonshot, so many of the people who had less resources, rural Appalachian people, people of color, they weren't really involved in that. And a lot of them were walking around in a jungle, 5,000 miles away while man was walking on the moon. And so, we have to think about, given our position just shy of Appalachia, how we can reach out to communities, because right now what we understand about the human connectome, and especially its development, is really the development of university professors' kids' brains, because those are the ones that are recruited, despite NIH spending $50 million to recruit a diverse population-based sample. When you look at the sample, you can almost recognize your colleagues from the connectome. And this is not an exaggeration. And I think that we have to think very seriously not only about being able to have a place, a home base, Houston, if you will, but a satellite that can go out and find families and do imaging. And we can do EEG, and we can do imaging, and we can collect biological samples, and we can do all of this, in a magic bus, essentially. And I can tell you more about that, but we're at that stage now, and then we can bring precision medicine and concierge care to people that don't have a child psychiatrist or a neurologist within 100 miles, and who aren't going to be able to come to us and really be seen in this fantastic new building on a regular basis. And in doing so, I think we link up to a lot of the other efforts we want to make in trying to be a good university, because one way to bring about democracy would be to change the way a whole set of states in the country thinks about science, for example, neuroscience. And we have the tools to make that impact.
Micah can speak to that.
Yeah, I was just going to sort of riff on that comment, Kevin. I think that's so important, and the work that we've begun here at UVA is very community engaged, and I think that needs to be at the heart of what we're talking about. Reaching out to underserved communities, partnering with people on the spectrum and their families so that their voices are informing the work that we're doing and is part of what's driving us forward. So I completely agree with those thoughts.
So Adema, if I know the gene, and why can't I predict what the mouse model will do if I know where it is on the synapse? What's the problem? Why can't I just do gene therapy and cure this kid of autism? Why doesn't that work?
Genes, much like brain itself, they don't act during their entire lives, during our entire lives. A gene is needed at a certain time, during a certain critical window. It may be needed later, but it might be that it's needed in a completely different function. The gene that I'm working on, it codes for a protein that is required early embryonically to guide, what Sarah was talking about, to guide neurons to their final location. And then it shuts down the expression. It doesn't pop up until, for example, eye opening in mice. And that tells us that it has these really distinct time points when it guides circuit development. And this is why I know that this gene is mutated in a subset of autistic patients, but I know that if I replace it, it will do nothing because it's way past its window. And this is why we can't do it. It's very simple.
Two things in terms of, again, fleshing out what we're thinking about in terms of moonshots. We really recognize that there's at least 2 periods of extreme plasticity that are important to understand. And obviously one of them is very early development. I think people are often stunned when they ask me about environmental causes of autism, and I say that, you know, the number 1 environmental cause of autism is the NICU. And they look at me like, are you crazy? And so let me parse that. We are very good, my colleagues like Karen Fairchild are very good at keeping babies alive and helping them through this experience and getting them to that next step. But we know almost nothing about how to optimize neurodevelopmental outcomes for those babies. And so that is a prime opportunity. Again, it needs the backdrop of an understanding of non-NICU baby development as well. But that's a perfect opportunity. And another one is, you know, why we're sitting in the rotunda. Adolescence is another period of incredible plasticity, and evolutionary theorists think that the reason why humans are so smart, and I know this is hard to remember when you're the parent of a teenager, but is because we have such an elongated adolescence. And we're just beginning to understand the neuroscience of adolescence. And so here I think autism is another extreme example where we can understand atypical brain development in a group of adolescents that's different, much in the way Rick Lifton studied, president of Rockefeller University now, but he's a geneticist who studied and figured out, you know, what causes some people to be very sensitive to salt by studying people who could eat all the salt they want, or, you know, couldn't go near salt and end up with either never changing their blood pressure or no matter what, if they touched salt, they had, you know, high blood pressure. And so autism is an example of an extreme form of adolescent brain development because many of the symptoms change and emerge. It's a period where we often see a second spike in, no pun intended, in cases of epilepsy. And so I think we had a hard time as a group deciding where to focus our grand challenge because both are so incredibly interesting, but I don't think I kept arguing that the principles are going to be very similar, and so no need to focus in on one.
So what kinds of investments would it take to achieve these grand challenges? So Micah, you can go first.
I think infrastructure to support the data science aspect of things, personnel to do the work, you know, new investigators who bring new ideas, I think would be the key aspects of this.
Jack?
Yeah, thank you.
You just want more bigger computers, but I wouldn't let you say that.
I'm always very sensitive to return on investment. The university is going to make a big investment here, probably in one of the things we're going to hear about today. The return on that investment is going to, if we spend that money wisely, we are going to position ourselves to be optimally competitive for major NIH initiatives. These large U54 class grants, which are multi-million-dollar awards, we'll be very competitive for. I think this autism spectrum disorder as an interesting test bed, bringing together multiple different data types, bringing together a team science approach, bringing together the people with the tools and talent and the technologies that we can bring together would be wonderful to kind of act as a little seed. And from that seed can grow all these different things. Micah talked about things like spinning off that could happen. These could be R01s. So I think this is an opportunity for the university to make a very strategic investment that will pay dividends in many, many many different ways. And so, thinking about people, thinking about tools, thinking about approaches and technologies will be very important as we move this particular moonshot forward.
Yeah, and I agree. I think that we keep bringing up this notion of a building, but one thing to emphasize is making the building modular so that as the science changes, it doesn't just become a place where people build silos and, and figure out, this is my lab and kind of business as usual, but that there's a natural built-in mix it up every few years, every 5 to 10, let's say 10 years or so. Just so thinking of it as something that's very modular. I mentioned the notion of truly implementing, going out into the community and collecting representative samples. As far as return on investment, and also both in terms of dollars and in reputation of the university, by going out into the community and doing things like what iThrive has been doing, hiring community-based interventionists and individuals working in the community, I think will be very important. And while hiring new colleagues is always very, very exciting and leads to lots of good dinners, I can think of 2 developmental cognitive neuroscientists right now, one that just got her K Award and is getting multiple offers from every school that was represented on that study section, to join them as tenure track faculty, but she doesn't have a tenure track position here. And I can think of a faculty member who has her first R01, and it wasn't when I got my first R01 when it was easy, it's now when it's hard. And she got it on her first try, and she doesn't have a tenure track position. And so I think taking care of our own talent would be incredibly important. And I don't want to get myself in trouble, but the next time a pandemic hits, the people that we value so much that work below the level of faculty, as faculty, we're all top 1% in the world of wage earners, every one of us in here. Maybe not in America, but in the world, we're all in the top 1%. But the people who still might make pretty good money, but are maybe working as nurses or psychologists implementing therapies, when they receive pay cuts because of a pandemic hitting, that really interrupts science and really destroys labs and lab cohesion. So I think we've got to start making up our own bed and taking care of our own room before we think about hiring scientists, from the outside. We've got amazing talent here already that we're just going to lose to UNC or Duke or Virginia Tech or Yale or Harvard or Stanford, just because we can't keep them on board.
Adema?
So,
and you have the last word, I think, 2 minutes before we open it up.
I'll be quick. I will echo a little bit what the previous panel mentioned of how difficult it is to work in development. Our lab works both with developing mice and with adult mice, and to address the developing mice and the questions that we're trying to really answer, we need better tools. A lot of tools that work for adult mice simply do not work in young mice. They're either not really working all that well, or they're too toxic. We just cannot really get to really dig deep into this question. So this is something that we would need. We would need tools and infrastructure and support to develop new tools and to develop new ways to approach these questions in the developing brain. Another thing that this panel touched upon and that I wanted to mention for us as scientists, what is really key is not just reaching out to the community, to those patients, to really incorporate that diversity, but also to make the community trust us again. If one thing, we've seen one thing over the last 2 years, and that's that People don't really trust scientists anymore. Everyone's doing their own research. And this has been detrimental for many, many reasons. And I think UVA is at a really prime position as the great and good to really reach out to those communities, to work with them bidirectionally, to have them trust us back for long-term investments, because those people are going to vote for people that will vote for investments in science. And this is kind of how we have to think about it. We have to think about it not just within the next 5 years, 10 years. We need to think more long-term. How can we really solidify scientific progress? Thank you.
All right. This is open for discussion. Questions? Comments?
So Harry Sondheimer, Neuroscience. I want to have one comment and then a question. The comment is that you guys done a phenomenal job in describing autism, but you know what's oftentimes is overlooked in the general population is that the vast majority of autistic individuals are actually low-functioning autistic individuals. And I think that the Rain Man movie actually has done more damage than good to the societal perception of the problem. As you know, I was directing an intellectual developmental disability research center at UAB in the Sparks Clinic, where we saw a lot of autistic kids. And I think that that actually has hurt science and has hurt investment in the problem. So that's my comment. And so I applaud you for marqueing autism as such an important problem. But that gets me to the question that the previous panel made a very eloquent argument for understanding normal development as well as abnormal development in the context of intellectual developmental disabilities. And many places would actually have autism be part of that same investment or same construct. And looking at Adema, I think your work dovetails very nicely with the developmental neuroscientists that I saw in the previous panel. Where do you see the synergism, and where do you see maybe ways to add to what they've presented in the previous panel?
I think I'll jump in first and maybe kick it to you, Kevin, or Adema. So I think we, as Kevin mentioned, we struggled with ironing out one of many potential moonshots. And so one of them is, in order to understand the mechanisms in early development and early predictors of autism, it does need to be more than just an autism sample, obviously. It would need to be very early longitudinal study of lots of kids and mapping out trajectories across them. So, Kevin, I don't know if you want to follow up on that idea or...
Yeah, no, I think absolutely. And we, as you know, the IDDRC is a very rarefied club. And a group of us, the last time the call for submissions came out, they made it clear they could fund up to about 3, and that 3 were up for renewal. UCLA, Harvard, and UNC, where I trained. And so, we nonetheless, it came out 30 days in advance of being due. So the other IDDRCs had worked on it for 5 years, and we got to work on it for 30 days. But we put together, one where we planned to study intellectual disabilities in multiple forms. Autism was certainly one of the ideas to focus upon, but it brought together immune function, microbiome, early neurodevelopment, and NICU as a particular model. And I think that's, and we got a great score. We weren't funded, but we were encouraged to come back in and come back in with more basic science. So I was delighted when you became the chair of neuroscience, having visited your center at Alabama. And so I think that we have, that's an example of return on investment, that the only real criticism of the proposal was that the university had not really put any effort into helping us get ready. We didn't have a LEND. We have a LEND now. So all the elements are there. So that would be like first on a list, and I think that in terms of comparisons between the prior panel, we are one. I mean, it's not, there's no, well, we might be two separate panels, we're all integrated and collaborating, and we'll go after all of these things together, and that.
Yeah, I just want to address the synergies question. So I'll make a counterpoint and say that synergies are wherever you find them. Put all of us in a room together, we will find collaborations. When I joined UVA, I had absolutely no expectations and no idea that, well, one day my lab will end up working with people. We will work with the NICU patients to try to improve their cognitive outcomes by developing new therapies. And for synapse neuroscientists like myself, that was impossible. But that simply came about by shared interests. I study preterm birth as a model of experience and as a model of traumatic experience early birth. And other people like Megan Pouliot study preterm birth as a cause of neurodevelopmental disorders. And this brought us together simply because we want to. And this is where the synergies will come about. Give us the opportunities, the synergies and the work together will arise. Teamwork is not that hard.
I think Provost McGill had a question. Questions, comments?
This may just be my desire for optimism. Many of you started with what we don't know, but we know more than we did 10 to 20 years ago. So maybe one or two of you could talk about what you're building on that were incredibly important breakthroughs. Kevin, you talked about the gender difference, which is extraordinary, and seems like very promising going forward as you subtypes and the sorts of things you were talking about, Micah. But I wonder if there are other things that your work all builds on now to give us a sense of where we came from, because we've learned a lot. We still don't know a lot, right?
Well, I think one of the things that, focusing on UVA work, but we're now, we've demonstrated and replicated that we're able to use brain imaging to predict which behavioral intervention will work for which kids and why. And so this opens, I think this is an important discovery because it opens the opportunity to then target the non-responders because of the answer of why. So for example, in one clinical trial that we have ongoing, we're using intranasal oxytocin to prime the brain to turn a non-responding child into a responder based on the neuroimaging data that we collect prior to the onset. And the reason why that's important is that if insurance isn't paying for it, in many states it doesn't, these are $30,000 to $60,000 courses of treatment, and they're 16 weeks long, most days of the week, and usually one parent has to not work, so who's paying for it if there's only one parent? And in addition, I see ways that we can work with our colleagues here. For example, on my to-do list is to sit down with Dr. Elias and talk about brain stimulation with the same sort of idea of turning a non-responder into a responder, and it requires that constant back and forth between clinicians. Like that study would only be possible with a collaboration like Micah and I have, and then Jack helping us do the machine learning that was necessary to do the neuroprediction. So that's one, I think, major breakthrough that was unheard of 10 years ago.
And I would just add that I think, as a field looking back a couple of decades, we certainly have made advancements in being able to do better assessments that are picking up on signs and symptoms and difficulties much earlier than we did before, and for people with more subtle difficulties. And we've developed interventions, not just for the core features of autism, but things like some of my work focuses on sleep interventions to help, because insomnia affects about 80% of people on the spectrum. And we found that if we can intervene and get children sleeping better, their cognitive functioning is better during the day, their behaviors and social interactions are better. So things like that, we're learning a lot about how symptoms affect one another. And I think by pulling in neuroscience and basic scientists, we could learn so much more about how to target those and understand who's going to respond best to which treatment and what to start first.
So I'm going to put John Lukens and AO on the spot. What do you guys think, immune therapies-wise, what's coming on autism? If you could...
Yeah, so I think, well, thanks for putting us on the spot again, and I'll carry us, AO, but if you're ready to jump in whenever. I mean, I'm coming to autism as an outsider, as somebody that works on the immune system. And, you know, one trend that I've really enjoyed seeing in the autism field is, you know, you look back 10 years ago and people were scared to study the role of inflammation in pregnancy. I mean, for various reasons which are probably clear to everybody. But that field has exploded in the last 5 years. I mean, it's kind of crazy. Like, I had trepidations too, of looking how inflammation that's associated with an infection alters the developing brain. And, you know, lucky enough, I was surrounded by people that said, no, you go for it. And that's what the strength of UVA has been. And I think of this year when we went through SARS-COVID, and the Simons Foundation, which is an amazing leader in the autism field, there was a call out to track mothers and neurodevelopmental disorders. And, you know, for everybody that's had children or have kids, when you do go for your checkups, you're always giving blood. You're always giving blood. And it would be really cool if they included an immune profile or a microbiome profile in that assessment, because that's where it starts. It really does. And the animal data, and it's not just mouse data, it's primate data too, shows that. I'll let AO jump in.
Well, since I'm forced to say something, I just want to emphasize the last thing that John said. COVID has come, it's here, the immune system is activated. We don't know where that's going to take us. So to the question of the immune system and autism, I mean, we can't back off now. Like we have to go there in light of what COVID has brought to us. And it's a challenge, but I think, again, UVA is a great place to do that.
Any other questions? So we have perfect time. Go ahead. Amanita.
This is Aminata again, sorry. I like Dr. Mazurek's comment about this needing longitudinal studies to see the outcome of possible therapies or the outcomes of challenges like immune challenges earlier in development. But oftentimes we know in science, getting funding for longitudinal studies is hard, right? Because you need to have things that are presentable today or in 2 years in order to keep funding going. So are they going to be mechanism in place with these grand challenges to help support labs and trainees to keep these things going so that we can get the desired result in 5-10 years?
So Ada's question is that if you want to follow a large cohort, whether it is in experimental animals or humans, over a period of time, that becomes extremely expensive. And can UVA's grand challenges support such an initiative? And briefly, my answer would be it could leverage some money from NIH to do it. We don't print money here, only Washington does. But I would ask Kevin and Jack and Micah to respond to that.
Well, I think that what we're arguing for is the need to build the initial foundation and the upon which to then develop this really beautiful platform where we can go after all sorts of NIH funding to support the longitudinal cohorts. Once we prove that we can do it, we can get a whole range, and then there's a point where in study sections, once you have done a longitudinal cohort long enough, they are very hesitant to kill it because they look at the Framingham study, for example. I mean, we might not have Ted Kennedy as our senator, but nonetheless, they didn't kill that. I think it's soaked up about $1 billion at this point of NIH funding.
I would agree with Kevin, is that this is providing that initial investment. It's something we can really build on. UVA is ideally positioned to bring together the people in this room, the people at this table, in a team science kind of approach to make ourselves maximally competitive for those big NIH awards like Kevin's alluding to. Can you imagine if we landed something like the Framingham kind of study and got that started and followed that, and we had that kind of resources and that kind of data and that kind of longitudinal component we could track over time in autism or developmental neuroscience? That'd be incredible. We'd have a license to print Nature and Science papers. It would be so exciting. And it's bringing together data across all these different spatial and temporal dimensions. And the NIH is very interested in those kind of programs. These U54 grants, which I kind of alluded to earlier, these are the big ticket items that really are multiple millions of dollars, multiple different institutions. We could partner with other institutions and position UVA at the top of the pile of leaders in neuroscience generally, as well as in development and in autism.
Yeah, I would just completely agree with what you've both said, and I think the key investments really are infrastructure in the form of people in a lot of ways to make this work happen, to go out into the community, to collect data, to have the specialized training to be able to do the things that we need to do to pull off something like that. And that infrastructure would support multiple iterations and I think would be competitive.
One last point, since we all brought up, or two of us brought up the Framingham study. So the Framingham study originally had two possible locations, Framingham or Paintsville, Kentucky. I'm actually from Paintsville, Kentucky. I think the only other person of any note is Loretta Lynn, and she's a lot more famous than I am. But obviously, Kentucky didn't have quite the political representation that Massachusetts had. And so Paintsville wasn't selected. But imagine if there were a university, there that really championed making that impact. I doubt the Framingham study, altered democracy as we understand it in Massachusetts. But had it been positioned, very close to the Appalachian Mountains, where we could build confidence in science again, and really change people's opinions, then I think that the great and good can be affected in ways that we hadn't really thought of by virtue of following longitudinal cohorts in places where they aren't usually followed.
I think we're coming to end of our time, and I see President Ryan has entered, so we'll stop. Thank you. Thank you so much to the panel.
Thank you, Jaideep, and the panelists. When President Ryan and Liz, Provost McGill, summoned Megan and I and said that now that seemed like the cloud on COVID-19 seems to break a little bit, what can we do to advance the strategic areas? And we should be thinking big, and it's what kind of challenges can we solve building on UVA's strengths. And as a result, the announcement that came out, Liz and Jim, as Liz talked about, it came out on the grand challenges, and this forum, all the way, the process has evolved. So it'll be a pleasure to have President Ryan share with us, in his own words, we can hear from him, his excitement for the grand challenges program. President Ryan.
Good afternoon, everyone. I am incredibly excited to be here. I wish I could be here all day, in part because although I'm completely a novice, the field of neuroscience is something that I've been interested in for quite some time. But also because this is an unusual situation. And before I explain why, I want to thank all of you for being here, thank the moderators and the panelists. Thanks to Liz McGill and to Ram, and as well as to Megan for organizing this. So this is, I don't know about you, but this is a very different kind of gathering. A lot of the meetings that I'm in are about how can we do more with less? How can we deal with constrained budgets or budgets that are being cut? Or how can we fundraise for things that we really want to do? It's a rare opportunity to be in a room where the question is, what could we do with $20 to $50 million? Which is why I wish I could spend all day here, because I normally don't get to spend my days thinking about something as exciting as that question. I want to give you some of the context of how we got here, and my guess is Liz talked a little bit about this, and I apologize if I'm just repeating what she said, but when we pulled together the strategic plan, investing in research was obviously something we wanted to do, but rather than spread the peanut butter as widely and thinly as possible, we thought we would have a better impact, a greater impact, if we focused on 5 areas. And those 5 areas are democracy, environmental resilience and sustainability, digital technology and society, precision medicine, and the brain and neuroscience. And we picked these 5 areas for several reasons. One is because each one of them is important to our democracy, important to society, important to human flourishing. But also because we had the strong sense that there will be important discoveries made in each of these areas in the next 10 to 15 years. And then third, there are areas where UVA would be building on strength, and building in particular on interdisciplinary strength. And so these are the areas that we thought we ought to, over a period of several years, make significant investments in. This is not to say that no other research matters or no other research is going to count, but in terms of university resources, we thought these are the 5 areas where we're going to put in significant resources. Not to support basic research, not to substitute for funding grants, but really to invest in infrastructure or invest in people, whether it's cluster hires or leaders in their field or postdocs or doctoral students. As far as brain and neuroscience, I mean, it fits all of the criteria. This is obviously an incredibly important area of study. In some ways, it is the frontier of life sciences. We are also, it seems to me, poised to make important discoveries over the next 10 to 15 years on a range of issues and topics, from Alzheimer's to Parkinson's to strokes to understanding more about human development, to understanding more about autism, to understanding more about learning disabilities. And it's an area, as those in the room are living proof, where we not only have great strengths, but a track record of collaborating across schools and across disciplines. So the STAR initiative is just one of a number of examples. And the Brain Institute has helped this enormously, I think, and I'm grateful both to Terry Sullivan, my predecessor, and Tom Katsileas, Liz's predecessor, for establishing the Brain Institute, as well as to the faculty who have participated in it. So in a sense, your task is a challenging one, but I hope an exciting and an energizing one, and that is really to help us decide what are the most important questions that we're poised to answer? What are the most important discoveries that we're poised to make? What are the most important puzzles that we are poised to solve? And then what are the resources necessary to enable us to answer those questions, to solve those puzzles, and to make those discoveries? So like I said, I hope this is an exciting and an energizing exercise for all of you. It's absolutely critical to us in terms of figuring out, honestly, how to make the very best investments that will lead to discoveries that will improve and save lives. And to me, this is a perfect example of what I mean when I talk about being both great and good, because the research that can occur in this field is not only exciting in terms of the pure science of it, but it also has the capacity to change countless lives. So thanks again to all of you for being here. Thanks for the work that you do, and thanks for letting me interrupt your conference. Thanks.